We are talking about DNA replication today. So, first we're going to start with the Human Genome Project, which, if you've ever seen any form of like sci-fi movie that they're talking about some form of DNA, you've probably seen one of these photos. The genome is going to be your complete set of genetic information and your genetic material found in an organism. This is basically going to be one big diagram, one big map to show where genes are on chromosomes. We can now sequence the human genome and see the sequence of nucleotides, which means they can break it down and see your exact base pairs. They can see how many thymine you have, how many adenine, guanine, cytosine. It's kind of creepy, but it helps in diagnosing diseases or illnesses. And then as science has progressed, this has helped in like prevention and potential like vaccines or stuff of that nature. And it kind of helps provide more insight into what we didn't know before so we can prevent a lot of things from happening. This is also really cool, and it's used in evolutionary studies. So basically, they can look at an embryo, which is just like a mass of cells, and kind of compare them from one organism to another to see how similar or different they are. So first, we're going to talk about DNA structure. So we know DNA is made up of nucleotides. This is going to be your sugar, your phosphate, and your nitrogen base. DNA is going to be made up specifically of deoxyribonucleic acid. Here is a diagram. The deoxyribose is a pentose sugar, so it's a 5-sided sugar. Your nitrogen bases are going to be what attach to the sugar. So your phosphate, as you can see here, is going to make up your backbone. Then it's going to connect to the sugar, and this is going to have an ester bond. And then your sugar is going to be connected to your nitrogen base. And it's just going to keep mounting on one side and then the same on the other. So we have two types of nitrogen bases. We have guanine and pyrimidine, or not guanine, I'm sorry, purines and pyrimidines. Purines are going to be double ringed, so they are bigger. A and G are going to be your purines. If you need to remember a way to like differentiate which one is the double ring and which one's the single ring, purine is the shorter name but the bigger base. Pyrimidine is the larger name but the smaller structure. Cytosine, thymine, and uracil are going to be your pyrimidine bases. Uracil is found only in in RNA, and uracil and the T, thymine, are going to swap out. So here are all of our bases. We've got our purines down here, so that double ring. Pyrimidines are up top. That's going to be your U, C, and T. So these base pairs have a partner that they are always going to find. G is always going to find C, and T is always going to find A. This is going to make them complementary. And Erwin Chargaff, he is a scientist. It's basically Chargaff's rule that G and C are always going to be the same, and T and A are going to be the same. With that being said, because C is always going to find G, they're always going to be the same amounts of that. So if you have 15 cytosines, cytosine is going to pair up with guanine, and so that means there's also going to be 15 guanines, okay? These are called base pairs. These base pairs are going to link up, and they're going to be connected through hydrogen bonds. So we talked about that when we learned about water, and together they're going to create this double helix structure. So here we have one side of a DNA strand. So this is like one nucleotide, another nucleotide, another nucleotide. And let's see if this works. So guanine is on one side. It's always going to match up with a hydrogen bond with cytosine. Same thing with thymine and adenine. Ooh, that's a weird circle, whatever. Uracil is only going to be found in RNA. So if you see a T in your DNA structure, like in your diagram, and you got to figure out if it's DNA or RNA, the T and the U are the automatic giveaways. Okay, so they're going to link up, and then together they will create this double helix structure that you should be familiar with. So Rosalind Franklin, James Watson, and Francis Crick, these are the scientists that really spearheaded it. In all honesty, Rosalind Franklin really got like um, the poop end of the stick, and she was dead. She had passed away by the time that they had won the Nobel Peace Prize, and based on the committee, those who are deceased cannot be recipients of the award. So she really got, left out in that one. But together they developed the double helix shape. DNA reads from 5' to 3'. So it's just the direction that it moves. And then from top to bottom, it's going to go 3 to 5. So let's go back real quick to this diagram. So here is 5' to 3'. So 5 is up here, and then 3 is down here. So this, it's always going to read opposite. So if this side is 5, so the G is on the 5 side, the C is going to be on the 3 side, okay? And then the same is going to be for the 5 on the bottom. So it's read differently. So you see how on this side, this deoxyribose sugar is backwards, the P, they're going in opposite directions, okay? And that's going to be called making them anti-parallel. So they are, parallel where they're basically running alongside each other, but they're going in opposite directions. So our chromosome structures, we should be pretty familiar with it because we have been talking about chromosomes for a while, especially like cells and cell cycle. So I kind of just wanted to tie it all in together. So we should be familiar with chromosomes. But basically your DNA is going to coil around a histone, which is this really tiny protein, and it's going to create a nucleosome. And then so here's your DNA, here's your base pairs. It's going to create your double helix. It's going to start wrapping around this histone, and then it's going to start coiling up to create your chromatin. Your chromatin is going to be coiled up again to create your chromosomes. So hopefully this kind of connects what we're doing now with what we've done already. DNA is going to replicate in a semi-conservative replication, and what this means is, so this blue up here is your original DNA, okay? And to replicate, this DNA is going to split, and then one side is going to form one round of replication, and the other side is going to do the other. So as you can see down here, your blue, which was our original, only makes up half of the DNA, and that new strand that it made is yellow. So semi-conservative means you're going to have one of the original strands of DNA and then one new one that's going to match up exactly to it. So during replication, DNA strands separate and serve as a template. You have one original strand of DNA and one new one. This is an interphase. Remember, we talked about when DNA is going to replicate. It's going to happen in the S phase. And then our 3 main stages during replication is going to be unwinding, base pairs, and joining. So your unwinding is going to be when that DNA is going to unwrap. Our base pairs is when we're going to match up the complementary pairs. And then joining is going to be making sure the strand is set and good to go. So unwinding, DNA helicase, which is an enzyme, it's responsible for unwinding the DNA and unzipping it. So think of it as a zipper on like your boots or pants, your jacket. It's literally coming up and separating the DNA. So it's basically going to cut up those hydrogen bonds. It's going to leave you with a single strand of DNA. And then your RNA primase, which is another enzyme, is going to come and start adding your primer to the DNA. So your origin of replication is going to be where your replication is starting. It's kind of the starting point, and your DNA is going to make its way all the way around and then end up right back there. Then our base pairing is going to happen when DNA polymerase, again, another enzyme, remember ASE means enzyme, is going to start adding the correct pairs. So every time, we're going to start with the 5'. When we see a G, we know G is going to match with C, so that's the base pair that DNA polymerase is going to add. G, when it sees the C, it's going to add the G. When it sees the A, it's going to add the T. And that's what DNA polymerase is going to do. It's just going to go up and put the correct partner together. So at our old DNA, we're starting at the 3' prime, but remember DNA is going to be read 3' to 5', and it's always replicated 5' to 3'. So we're going to have 2 strands because obviously you have a lot of DNA and this is going to take forever. So we have a leading strand, which means it's just going to do one very long continuous strand from the beginning of the origin of replication to right before the next origin of replication. And then you're going to have a lagging strand that kind of chunks it up a little bit. And it's going to chunk it up, and then we're going to have other enzymes come in and just kind of plug it all up and make sure it's okay. So proofreading, we want to make sure that our base pairs are correct and kind of eliminate all error that might have been made. DNA ligase is going to go through and it's going to start closing the gaps. So this is our leading strand. It's continuous and it's going to go all the way until it's done. The lagging strand is going to go in the opposite direction and it's going to chunk it up. But with these chunks, we need to make sure that they are sealed. So DNA ligase is going to come in and just kind of solidify everything to make sure it now becomes a continuous strand. And that's it for this PowerPoint.